Hello, and welcome to today's webinar. This is Christy Sullivan, and I'm the Secretary of the American Society for Cellular and Computational Toxicology. This webinar, like our series of monthly webinars, like all of them, is presented by the European Society for Toxicology in Vitro and ASCCT together. Both of our organizations host regular meetings and pursue other activities to promote computational and in vitro toxicology fields. Each organization offers training and education and also has programs to support early career scientists in their endeavors. First of all, some general housekeeping items. This webinar is recorded, and the recording will be posted on the ASCCT website, which you can see here on your screen, ascctalks.org, a few days from now. Everyone will also receive a PDF copy of our presenters' slides today. Also, the lines are muted. There will be an opportunity to ask questions after each presentation. We'll have a speaker, and then we'll have a Q&A period, and then the second speaker in a Q&A period. You can ask a question by typing it into the Q&A module on your GoToWebinar control panel, and I will be able to read out your question after the speaker is finished. If you have any technical issues, you can also reach me using the chat box during the webinar. Before we start, I want to just offer a couple of highlights of future activities from both of the societies. First of all, the ASCCT is having its 9th annual meeting in October, and it was planned to be in North Carolina, but we have decided to take the meeting online. So it will be completely virtual. We will still offer a full meeting with plenary speakers, many of which you can see here on your screen, as well as talks chosen from submitted abstracts, panel discussions, poster presentations, and opportunities for engagement and discussion. We hope you will be able to join us. Given the online format, maybe we'll be able to attract some more people who wouldn't normally be able to come to our annual meeting. And just to note that abstract submission is due August 7th. So if you want to be considered for a talk or a poster, please submit your abstract by August 7th. And here again, you can find out more information on the ascctalks.org website. And we have a few upcoming webinars in August, September, and October, and you'll be able to register on our website in a few weeks. And also just to note that the ESTIV annual meeting is going to be rescheduled in Barcelona in 2022. And they are having a training course in October of this year. You can find out more about that on their website, estiv.org. For today's webinar, we're going to be hearing about human biology-based approaches for COVID-19 therapeutics. The first presenter is Dr. Malcolm Wilkinson, Kirkstall Limited. Malcolm retired as CEO of Kirkstall Limited in 2019 and is now busier than ever helping research organizations and charities working to replace the use of animals. Previously in his career, he had senior roles in both research and development and sales and marketing. He founded a consulting company which supported spin-outs from universities and raised over $15 million from venture capital and regional development funds. He has been a visiting lecturer for FSRM in Neuchâtel, Switzerland on the subject of micro and nanotechnology in biomedical engineering for over 10 years. And in 2019 was appointed as visiting professor in the Department of Biological Engineering at Sheffield University. Dr. Wilkinson is co-author on several papers on in vitro models of toxicity and a contributing editor of a recently published book on in vitro testing. Our second speaker is Dr. Samuel Constant of Epithelix. Dr. Constant is co-founder and chief executive officer of Epithelix, a Swiss biotech company specialized in tissue engineering. Epithelix is a leader for in vitro assessment of drug efficacy and toxicity on the human respiratory tract Epithelex has developed unique 3D in vitro human airway tissues and testing services for studying airway pathologies like asthma, cystic fibrosis, and chronic obstructive pulmonary diseases, and bacterial and viral infections. Samuel is in charge of global management and business development of the company, and with external collaborators with private and public research groups. And with those introductions, I will go ahead and turn the floor over to Dr. Wilkinson for our first presentation.
And let's just see if I can get this started. Yeah.
Okay.
So,
yep, looks
yep.
good. Oh, go ahead. Thanks.
Great. Okay. So, I'm going to talk about really developing models, in vitro models, as a way of replacing animals, and that's my passion at the moment. As Christy said, I've retired from full-time employment, but I'm still very, very busy. So what I'm going to cover is first of all a little bit of introduction. So hang on a minute, I'm just trying to get rid of the. Can you see my control board on your screen, or is that muted, hidden from you, Christy?
It's hidden from us.
Okay, that's great. Yeah.
Move it. You can hit the.
Yeah. Okay. Yeah, it's okay. As long as you can see the slides, I'm okay. So a little bit of background, because it's why do people use animals? If we're going to replace animals, we need to know, absolutely be clear why people are using animals. I've also then just got a little bit of a background on what industry is looking for in terms of replacement models. And then I'm going to give a little bit of the roadmap of how the in vitro technology is developing. Academic research is absolutely crucial to the introduction of new technology, and they tend to be willing to try out new methods. And so I'm delighted that the company that I was working for, Kirkstall, has agreed to support one of the leading charities in the UK, that's Animal Free Research UK, and we're going to be supporting some academic projects. And the second half of my presentation is going to be a little bit of detail about the first 4 projects which have been approved and supported by Animal Free Research. Okay, get this to progress forward. Yeah, there we go. So why do people use animals? And if we're going to replace animals by in vitro, what do we need to be achieving with the in vitro methods? Well, I think the big attraction of animals is it's a complete systemic model. You've got the whole organism there with liver and kidney and everything working together, and you can metabolize drugs and chemicals. The other thing about the animals is that you can run experiments over long periods, and some animal tests actually go on for 90 days, which is much longer than general in vitro tests. And if you feed the animals properly, look after them, they maintain homeostasis, and before you start giving them some toxic chemical, at least they are healthy. The interesting question is, how can you use animals to model human disease? Because many of the human diseases and, for example, the response to the COVID virus isn't seen in animals. And of course, there is a school of thought that says, oh, well, you can modify the genetics of mice and give them a susceptibility to diabetes or some other disease, but really they're not going to be human, whatever you try and do. So meanwhile, we're really been developing the in vitro models, and it's been a very exciting ride, because over the last 50 years or so, we've gone from very simple 2-dimensional cell cultures growing in glass plates to high throughput systems, 3D culture, and then adding more physiologically relevant factors like the flow of media, and then co-culturing different types of cells. And of course, the excitement at the moment is about organ on a chip. Can we actually build organs on a chip or humans on a chip? But I'm not going to talk about that today, although it is a particular subject of interest for me. So meanwhile, let's just look at how industry views the changing scene of in vitro work. This is a slide from AstraZeneca, and it shows very clearly how they see the two-dimensional culture evolving as you go from left to right into multi-organoid type cultures. And it's quite clear that the more complex the model, the more information you may get. But there is a trade-off for industry, because every model, as you go more complex, is more difficult to run. It may become more relevant to the human in vivo system, but your throughput will be dropping off. And of course, for many applications where you're trying to screen large numbers of compounds, then throughput is very important. But for animals, of course, we're not using them for high throughput screening. So we can think about more complex models. Now, as I said, the animal research or replacement of animals is now starting to really become a hot topic in universities, and universities are, I think, going to be using a lot and developing a lot of new techniques. And so Animal Free Research UK, which says the leading charity in the UK, and the company that I used to work for, Kirkstall, got together, and they put out a call for proposals about 3 months ago, and we got a very, very good response to that, some wonderful projects. And all these are aimed at not using animals, not using animal materials. And the technical panel, the scientific advisory panel of the charity, has selected 4 projects, which will then be funded by cash from Animal Free Research UK, but also get free equipment from Kirkstall Company. And the 4 projects which have been approved, just getting started, these are between 8 and 12 months long. They're generally feasibility studies, initial starting projects using new technology. And so we've got 3 in the UK, but we've also got one from, of course, John's very well known in the animal replacement field. And all of these really have the objective of building better models to try and understand respiratory disease, particularly COVID-19, using human cells and human materials. And so what I'm going to just mention is why these particular groups have selected the Quasi Vivo as a tool to help them do that. So for those of you who are not familiar with Quasi Vivo, it's a very simple cell culture technology, modular. It's a little bit like Lego kit for cell biologists. You have various building blocks. You have a reservoir bottle in which you keep your media, and there'll be sufficient media in there to run an experiment for maybe 3 to 7 days. The media is recirculating because it's pumped around the system, and it flows through individual chambers, and this is where you culture your cells, either at the bottom of the chamber on a glass cover slip or plastic, or on a scaffold, or even put tissue slices into these chambers. And you need a pump to gently move the media around, and the flows we're talking about tend to be very, very small flows. They're more modeling the sort of interstitial flow of media through the microvascular rather than the arterial flow. So very simple system. That's the components that these research groups are going to be using in their models. And these are all commercially available. So we have a number of different types of chambers and a number of different sorts of pumps that have been designed. But the key one really to look at is the one in the middle there, which is the QV600, because that was designed specifically to handle the air-liquid interface and respiratory tissue using standard transwells. So those are the tools, and the idea being that the flow of media has a lot of beneficial effects. It's not only delivering media and oxygen to the cells, it's removing waste products, but strangely, it's the flow of media actually stimulates the cells. And we got lots of evidence that actually cells are happier under flow than they are under static conditions. And of course, once you've got flow, you can connect together different chambers and look to cell-to-cell or tissue-to-tissue signaling, and you move towards multi-organ models. So those are the sort of technical features which we hope are going to give benefits to these models, these research projects. Now, the data we had previously mainly was on liver, but more recently on lung and barrier models. And just show you some of the data here very quickly on liver, because surprisingly, liver is quite a difficult culture to keep active, and human hepatocytes actually function very, very well under these gentle flow conditions. So you know, not only get gene expression, you get activity, and you can use that culture then to predict the toxicity of drugs. So it's a bit of an aside, but the evidence is there, a lot of published papers on that particular benefit of this system. So what we're really interested in today is the model on the left-hand side. Most of the projects are going to be using a lung model, and they'll be taking a standard transwell, which is shown in the bottom little diagram there, with air above the cell culture and liquid media on the basal side of the insert. And that system actually is running in this QV600 chamber. On the right-hand side, we've got a more complicated model, but that's not really relevant to these first four projects. So the question then is, given the flow of the media past the bottom of the basal side of the transwell, what's going to happen? Well, the flow, strangely, will actually stimulate the cells in that epithelial barrier, and the barrier function improves and viability in such a thing. So that's what we're hoping to see. You can also put the virus into the media, the flowing liquid, and see if the cells in the transwell will be affected. So those are the sort of experiments that people are going to run. So let's look now at the first four projects which have been approved, and most of these are just getting started, so I've no data to show you yet, but what I'm going to share with you is the objectives and how the projects are hoping to improve the in vitro models. So the first one is from a group at Imperial College. The principal researcher there will be Carolina Herrera, and she's looking at, again, the importance of the flow of lymph and blood, and interacts with cells from the respiratory tract. The whole objective here is to end up with a physiologically relevant model that can be then used to test drug candidates and see whether they can inhibit the first steps of infection and then the inflammation process that starts. So very clear objective for the project, and clearly the flow properties of the Quasi Vivo system are crucial in this sort of experiment, because they want to have a liquid flux, the media flowing past, and they're going to not only sort of try and replicate the way that the virus comes from the blood into the system, into the lungs, but also look at the activity of the cells, cytokine secretion, going back into the blood from the infected cells. So those are the simple objectives of that experiment. On to project 2. So this is Mark Turner's the principal researcher in that project, and he's at Coventry University in the UK. And that one is interesting in that the data suggests that people who are obese are more at risk to developing severe symptoms from the SARS-CoV virus, and trying to understand why that may be. And the Coventry group have drawn attention to the the inability of animal models to really be useful because the metabolism is just so different from humans. So we need to be using human cells and a human tissue-based model for these systems. And the group are going to actually build a multi-tissue model using multiple chambers with the Quasi Vivo connected system. And I think it will be a general model, not just for the SARS-CoV, but perhaps a more wider application. Move on to what they're hoping to get in terms of benefits. Well, clearly by growing multiple different tissues and connecting them by flow, that gives them an ability to build quite a complex model. And again, using human cells, they'd like to build models which can just replace the use of animals in obesity and type 2 diabetes research. Once you've got that in vitro model, of course, hopefully you can scale it up and translate any new methods, assays from the test bench, the research bench, into a higher throughput test. So if you like, the initial Lego kit is a proof of feasibility, but what will be interesting is with any of these models that are successful, we aim to scale them up so you could have more multiple chambers in parallel, and really having a good model that you can mimic what happens in the body, but also scale up the number of tests you can run. Third project, this is Johns Hopkins, so that's over in Baltimore, and Vaishaki Ghosh is the principal investigator on that one. And here again, it's the lung model that's under study, and here it's the insight that the lung tissue is affected by smoking and so on, so that it then becomes more susceptible to viral infection. So its ability to combine all the knowledge that Johns Hopkins have got on the sort of cigarette smoke and vaping model that they've already done, and then combine that with the system that will look at how the COVID virus is then picked up by these this model. So why the what they're hoping to achieve with the this new system is it's to actually mimic the right phenotype of the epithelial cells with their smoke inhalation, and then use those cells as a target for infection by the SARS virus. And hopefully they'll be able to build that more complicated model, and then use it to see how therapies or drugs could actually prevent the infection or reduce its effects. So final project, this is from Liverpool School of Tropical Medicine, Samantha Donnellan, principal investigator on that one. And again, this is moving to a more complicated model again with two diseases, two human respiratory pathogens, both affecting the lung. So that's tuberculosis and COVID. And it's the ability to model diseases. So here they're hoping to get some preliminary data so they can quantify those susceptibilities and interactions. And then with the model, if it's successful, use that again for drug screening and more general application. So what we have there then is they're using a different Kirkstall Quasi Vivo system. That's the 900, because that one allows you to run 6 cells, 6 chambers in parallel, and it's geared towards sort of higher throughput screening. And it's also a system that's designed to allow live cell imaging while you've got flow of media running. So there's a couple of features there they've picked up on in their experiments. So I think we've got some very, very exciting projects there. As I said, it's very early days. No data yet, and I'm hoping that maybe in a future conference or webinar we can show later on how these experiments have generated interesting data. And I think for the animal charity and the animal replacement, we're also hoping that all of the techniques that are developed will have more general application than just COVID. Obviously, that's the motivation for this this particular project, but I think in general these models could well be much wider application and help us move in vitro tests and assays into the market as a replacement for animals. I think just like to round up by acknowledging, say, the support not only of the charity but of the company Kirkstall, who've provided the equipment to these different research groups. I'm certainly willing to take questions, although I think in many cases I may end up deferring those off to the researchers because these projects are really at a technical level, I think, where you need to be talking to the experts in the biology rather than myself. And I think of myself as a high-tech plumber providing these flow systems and so on, rather than a biologist. I think the charity work is support for these groups is brilliant, and I think you've got contacts there for Carla Owen and the charity group, and then Kirkstall, you've got to contact Mark Vo, who's the new chief executive who took over from me at Kirkstall, and the final contact there is myself. TFI is my own consulting company, and I'm busy, say, very active with a number of projects to try and develop a replacement for animals. So thank you very much for listening, and I'll do what I can to answer your questions. Kirsty, back over to you.
Great, thank you very much, Malcolm. That was a really nice overview of some of the technologies and projects going on. So we have a question already, and so just to remind everyone, you can enter your questions right now in the question module, and I'll read them out for you. So it's a bit long, so bear with me, but it's a good one. Great lecture elaborating on the projects and also linked to the intro on COVID-19 and role of endothelial cells. In relation to the internal barrier related microphysiological systems, a recent paper looked at advanced 3D microfluidic models of the human blood-brain barrier, a platform that most closely resembles the human physiological condition, showed the ability for SARS-CoV-2 spike proteins to trigger a pro-inflammatory response on brain endothelial cells that may contribute to an altered state of blood-brain barrier function. These results are the first to show the direct impact the SARS spike protein could have on brain endothelial cells, potentially offering a plausible explanation for neurological consequences seen in COVID-19 patients. So the role of endothelial cells seems critical. Do the projects that you presented include endothelial cells, which seem to be a critical part of the response.
Okay, well, yeah, if I can come back on that then, Christy. First of all, the choice of particular cells in each project is different. So I'm going to sort of hesitate to respond to that. I mean, the idea of trying to form a really good barrier is I think crucial, not just to the lung function, the airways, respiratory tract, but also to blood-brain, to the gut, and so on. And I think what's interesting in our experience is the blood-brain barrier has been one of those barriers that we've been collaborating with different academic groups, and it's clear that the flow in these sort of systems stimulates the creation of a much more robust, stronger barrier, measured by simple techniques like tear trans epithelial electrical resistance. But I think the answer is yes, barriers are very, very important, and I would hope that the cells that you can put onto these transwells, you can create these barrier functions and actually monitor how well they're functioning. If you are looking at how things are transmitted through a barrier, then of course there are other configurations. And I think right at the introduction, I showed a sort of liquid type barrier model for the blood-brain, which is one of the ones that's being used by other academic groups. So I'm sort of hedging my answer because I'm not an expert on this particular cell lines and cells that are being chosen, but I think the barrier function is very, very important to the this particular disease and many others.
Agreed. So the next question was also a compliment. Thank you. Excited to see how these projects progress. If the argument, so kind of hearkening back to your earlier slides, if the argument for using animals is that they provide systemic responses, how do we improve, or what else is needed to enhance the in vitro system to counter this argument and enable full replacement of animals?
Yeah, that's a really good question. So I think the first thing is that the in vitro models need to get more complex. So it's no good looking at just one cell type. If you've got, for example, one organ like a liver, you need hepatocytes, stellate cells, Kupffer cells, and so on. So we've got to move to more complex models. And then, of course, if you're looking at so many diseases, they're actually involve the interaction between different organs. So maybe the liver and the kidney or the spleen or whatever. And if you want the immune response, you know, you've got to have much more complicated models. But those are more tricky to run, you know, experimentally. So our research teams, our academics have got to figure out what are the protocols for building these more complicated models in a robust way that we can, you know, run in different labs and get reproducible, robust results. So I think it's part of a pathway that says we have to get more complicated models, but we're a long way from building, you know, organs, and we're certainly a long way from building connected organs. But we're on the right road, I think, and not just the Kirkstall system, but there's some excellent work going on in some of the other labs around the world, you know, tissues and so on in Germany and in Emulate in USA. These people are looking at that same challenge. How do we build connected, more complicated systems? So that's the road forward for our research teams.
Yeah, I also think part of this is knowing when you need that connection and when you don't necessarily. As you say, sometimes these are very complex, and so sometimes something more simplistic that gives us a sort of a screening answer is needed,
Yeah.
or combining the data from models rather than combining the models, if you will.
Yeah. Yep.
So next question.
Well, of course, it's
sorry, go ahead.
So, well, I was just going to say that we're talking here, I'm talking about in vitro, but of course, computer models, in silico models, and as you say, combining data and building simulations, if you like, are also part of the route to alternatives to animals. So, yeah, that's very important.
Great point. Can the cells in Quasi Vivo be in 2D monolayer or 3D structure? So either 2D or 3D. And then follow-on question, can Quasi Vivo work with multiple cell types?
Okay, well, the answer to both those is yes, but let's give you some examples. I mean, the idea of a 2D moving to 3D, the first thing that happens if you put more cells or tissue slices in cell culture is that you need to get the media and the oxygen into the depth of the 3D structure. So, for example, spheroids was one of the first techniques to try and grow more representative 3D structures. And what happens is if the spheroids get more than about 300 micron diameter, you get necrosis in the middle because of the lack of oxygen and nutrients. But flow helps you to culture more functional or viable large thicknesses of tissue. So the answer is you can do 2D and you can grow 3D and use tissue, right up to much thicker layers than you would without flow. In terms of multiple cells, now the interesting thing here is, of course, everybody has their own recipe for what is the best media for their particular cell. And I suppose the assumption is if you're going to culture multiple cells, how on earth are you going to find a media with the right growth factors or whatever in it that will suit all of them? And the surprise is that actually it's easier than you might think, because cells condition the media, and particularly once you have multiple cell types working together, they signal to each other, and we've got quite a lot of evidence from the different university groups using our system that happens. So yep, I think the answer to both those questions was yes, and there are a lot of published papers now showing the details of how these techniques have been set up in different research groups.
Great. Is there a risk of compound binding to the tubing of the system? So this is the Quasi Vivo system.
Absolutely, that's a good question, because that's one of the things that we found very early on, was that a lot of labs use PDMS silicones, and a lot of the chemicals bind, whether it's a drug or other materials, bind to these silicones. And the important thing there is to be able to model and measure how much binding is going on and have alternatives. So, for example, the choice of the plastic material that we use, we have different chamber materials in Quasi Vivo. We have acrylics and polymers. PDMS is one of the ones we use. And tubing, you can use, for example, Teflon tubing. It's not as simple, easy to use as silicone, but its binding properties are very, very good. And there was an excellent study done in Carolina by the Hamner Labs a couple of years back on binding in these different tubing. So I think the answer is yes, it's a risk, and you can mitigate it by choosing the different chamber materials or the different tubing materials if it's a problem.
Okay, great. Well, thanks for all of your questions, everyone. We're going to move on to the next presenter, Dr. Sam Constant. And so let me just give him the sharing slides prompt, and we'll go on to his presentation. Okay, I can see your slides.
Hello, everyone.
That looks good. Please go ahead.
Can you hear me?
Yep, we can hear you.
Yes, thank you. Thank you. Hello everyone. Yeah, it's a real pleasure to be with you today, at least virtually. So first of all, before starting, I would like to warmly thank Christy and Elizabeth for giving me this opportunity to present you, let's say, latest advance in the use of 3D human airway models to study SARS-CoV-2 pathogenesis. But before starting on this topic, I would like to remind you with some facts about COVID-19. Well, we have to recognize that, let's say, it's really a young disease in terms of research, because the first case were recognized only in December 2019 in China. And since then, a lot has been done in terms of research. And we can mention, for example, already in January 2020, meaning that only 1 month after the first cases, we had already the genome which was revealed and published. And then the same month, we had already also the first PCR diagnostic which was published. Then there were some key steps, like in March of this year, for example, the antibody tests were also reported. And in May, and this is really a big step, there was the first antiviral authorizations, which was given by FDA for emergency use of remdesivir, which was formerly used as a broad antiviral active compounds against several RNA viruses initially, and for example for Ebola. But now it was used for in the context of COVID-19 pathologies. So, and what is the status now? So, according to WHO website, so I've picked this data yesterday. So there are more than 40,000 research articles published on COVID-19 or related to COVID-19 research. So this is really huge. But also, and this is really the worst part, there is more than 11 million of cases reported, meaning that people were infected by this virus. And it is also intriguing to see that, you know, while it has taken 3 months to reach 1 million of cases worldwide, it has taken only 8 days to let's say have 1 million infected during the last past week. You know, so this is really an increasing, unfortunately, pathologies, which lead today in turn to more than 540,000 deaths worldwide. Okay. So how did that happen? Initially, so it has been hypothesized that let's say it was initially brought by bats, since we have found an homology of the virus genes between the human and the bat being approximately 80% homology. So it has been hypothesized that the bat has transmitted this virus to another mammal species, which is a pangolin. And here the homology in terms of genes is around 92%. And then from the pangolin to the human, and from the human to the humans, due to let's say the presence of the virus in the air and droplets, which are further on, let's say, absorbed through inhalation. Okay. So SARS-CoV-2 is a consequence of one virus, which is known, and the name is severe acute respiratory syndrome coronavirus 2. So this is SARS-CoV-2. Okay. So this virus has the following criteria. So the variant, which is depicted here, has a diameter of approximately something between 50 to 200 nanometers in terms of particle size distributions. So it is classified as a viral envelope, meaning that it has an envelope here, which is composed of mainly 3 types of protein, S, E, and M proteins, which protect the RNA genome, which is held in the center by N protein. It's also interesting to see this kind of spike here, which is known about S protein spike. Okay, this protein is really important because this is where it allows the virus to attach to and to fuse with the membrane of a host cell. So the chronology of events during SARS-CoV-2 is depicted in several papers, and I really recommend you to read the paper from Tai and Hal recently published, where they have let's say synthesized the different events caused by these infections. So first, it's present SARS-CoV-2 in the air and in some droplets, and the first portal of entry of this virus is the airway epithelium, meaning that when you breathe this droplet, it will touch the airway epithelial cells, and then it will be linked to the surface of the receptor of these cells. And the receptor for this SARS-CoV-2 is angiotensin converting enzyme 2, ACE2, present on the surface of the cells here. But it's not sufficient because the spike proteins need to be cleaved in order to enter and to fuse and enter into the cells. And it needs to have, let's say, protease, which is known to be transmembrane protease serine 2. And once parameters here active, then you could have the virus, which unfortunately enter into the cells and then release is let's say genetic informations and undergo virus replications, and the viral RNA will in turn form some proteins which could mature again to reform some variants which could be released from the airway epithelial cells apically, and then spread out to neighbor and infect neighboring cells. But also, as a consequence into the cells, there is a phenomena which occurs, which is known as paroptosis, which induces here, for example, some cytokines such as IL-6, MCP1, IP1, for example. And the role of these cytokines is to attract, let's say, immune cells, so such as T cells, monocytes, and macrophages, for examples, in order to protect the cells from, let's say, foreign, let's say, or danger signals. And it leads to, in a good healthy immune response, in good condition, if you wish, that the macrophages would, for example, clear, let's say, infected cells rapidly. But also, macrophages could also neutralize the virus, and using, for example, neutralizing antibodies here to, let's say, block the infectivity of the virus. And if everything is okay, it would lead to a minimal inflammations and non-cleavage. But in the worst case scenario, we end up with this situation, that we have a dysfunctional immune response. In this case, we can see excessive infiltration of monocyte, for example, macrophages and T cells, which would lead to systemic cytokine storm. Okay, and in the worst case, pulmonary edema, pneumonia are found, and widespread inflammations and multi-organ damage in the most severe cases. So clearly, the epithelia, as a human epithelium, has a large response in all these signaling pathways. So how do we model that in vitro? So we have initially taken advantage of the models that we have developed at Epithetics, human airway epithelium from the nose, trachea, and bronchi, with a mucillar system, which is made of 3 types of cells: basal cells, ciliated cells, goblet cells, and presence of mucus and cilia on top. And also small airway models. So this is the bronchial regions, where here we have another type of cells which come into play, which are known to be club cells, cuboidal club cells, or formerly described as Clara cells. So how do we reconstitute these tissues? Well, we start from human biopsy, and the process is really similar, irrelevant of the let's say location of the anatomic location of the initial biopsy. So we can start with nasal cells, for example, or tracheal cells, or bronchial cells, or small airway cells. We first have an amplification step, but not a large amplification, only one passage to be as close as possible to the native tissues. And then we seed these cells into microporous filters, transwell systems, and we switch the cells to the air liquid interface and let the system differentiate for several weeks, up to 6 weeks, for example. And we end up with a pseudostratified airway epithelium in all these cases. Pseudostratified means that, like in vivo, all the cells touch the laminar basis. And as you could see here, clearly, we can up with these 3 types of cells for the mucilary systems, so the same type of cells for the nasal, tracheal, and bronchial, so which are basal, ciliated, and goblet cells. And here we end up with models bearing this not only let's say ciliated and a few goblet cells, but here also club cells. And as you could see, like in vivo, the deeper you go into the respiratory tract, the thinner is the epithelium. So these models have been extensively used within the past 15 years for antiviral research, and in the context of, let's say, modeling, for example, the effect of rhinoviruses, influenza, but also enteroviruses, respiratory syncytial viruses models, but also they have been used for modeling metapneumovirus, parainfluenza. And finally, coronaviruses, initially with OC43, but we now have transposed these models to SARS-CoV-2, and I will discuss it in the coming slide. But before, how were they, let's say, efficient, and why do we use them? But first, the first thing was we have an efficient replication of these respiratory viruses on these models. And even very difficult to grow viruses which could not be replicated, for example, in some animals or in some cell lines, could be, let's say, very easily replicated in this non-modified human primary airway epithelium, such as, for example, rhinovirus C15, and they could be replicated, replicating for really long periods. This is an example for a 28-day study. So you could use them and wash off the apical surface to collect the virus, and they could be used as a producer, let's say an in vitro bioreactor to produce this virus to better understand them. So viruses are also known to perturb the host, and one of the most, let's say, important impairments is the mucociliary functions. So in vivo, in the human epithelium, you have cilia which are moving and which are, let's say, inducing a mucociliary escalator function constantly. And this is, let's say, replicated also in vitro on the mucociliary systems, for example, where you have a synchronized movement of cilia, and if you add some particles on top, you can track the movement of these particles with time, and it leads up to let's say a movement of the particles that could be quantitatively analyzed, and this is a condition which are non-infected. But here it is really intriguing to see that most of the viruses, let's say respiratory viruses, are interfering with this mucociliary function, and but not all, because some of them, like for example rhinovirus B or coronavirus OC43, do not interfere with cilia beating, with mucociliary clearance. So you are in this kind of phenomena. While you are here, you could see very clearly that there is no movement at all for other viruses. So this is a very interesting and physiological endpoint. So coming back to SARS-CoV-2, I've talking about ACE2 being, let's say, the receptor of SARS-CoV-2. So it is important to understand if it is present in the models. And indeed, this was the work, let's say, done by Sekana and Ugent University, where they have shown that indeed, ACE2 is very highly expressed at the mRNA level in the, let's say, the mucillary nasal or mucillary bronchial or in the small airway models. So clearly, this was the first basis that here they are clearly very well present. But more, let's say more convincing either are let's say the data reported here, where you could infect here apically with SARS-CoV-2 the models, and then follow the kinetic of virus growth with time. So, and this is a paper let's say published by Virpa's groups recently, where you could see that very clearly the nasal and bronchial epithelium could be used here to monitor, let's say, the of the SARS-CoV-2 up to, for example, here 96 hours. But very interestingly, you could see that the transepithelial reticle resistance is it's modulated by the presence of this virus. Also, it's very clear that in all the conditions, the tissues are still tight, meaning that let's say the value here are higher than 100 ohm per centimeter square. However, there is a clear tendency here of let's say decreasing, might be due to activations of ion channels. Okay. This model could also serve as a basis to visualize cell tropism. So these pictures, which are presented here, are electron microscopy picture, where you could see the presence of packs of viruses. So here represented by these arrows in some vesicles here, present inside the ciliated cells, but also inside the goblet cells. So this is very intriguing to see that SARS-CoV-2 infects mainly these two types of cells, ciliated and goblet cells, and to a lesser extent the progenitor basal cells. Also, the human airway epithelium is not only a physical barrier, but it's also a highly potent immunoregulator. And in this study, it has been shown that using a nanostring technology, that innate immune transcriptional signature are let's say induced by SARS-CoV-2 on mucillary. So a panel of genes have been let's say studied at different time points, and all in one, this data concludes that a strong upregulation of type 1 and type 3 interference are observed upon, let's say, a challenge with SARS-CoV-2, but also genes associated with NF-kappa B and TNF-alpha are also upregulated, which also validate also these symptoms, which are found also in clinics, also in human with early sign of the cytokine storm. So last here is with the fact that how can we use them for antiviral evaluation. So this is we need to find now some drugs, novel therapies or combos to fight against SARS-CoV-2. So first, this type of model being tight, being a DR liquid interface, is used to test different application modes, meaning that we could test systemic deliveries of, let's say, a novel therapy, adding, let's say, the compounds therapies or the combo in the basolateral medium, but also we could test also aerosol therapies directly on top of the airway epithelium, such as liquids, solids, nanoparticles, gases, or using a nebulizer for examples. Now, in terms of endpoints, in this strategy, we develop a multiple endpoint testing strategy with information from the apical side, being here the quantification of viral genome copy number. Here we can wash off the mucus and then extract the RNA to quantify this viral genome, and also to visualize if they are still infectious using TCID50 measurement infectious titers. We also monitor the effect on the tissue integrity with the transepithelial resistance, and from the tissue we could extract the, let's say, genes. So we can extract the mRNA to quantify the genes expressions and perform electron microscopy, for examples. And obviously, from the culture medium, secretion of soluble factors are really important, such as detections of cytokines, chemokines, and metalloproteinase. So here are data that I'm happy to present here, the way we use them for antiviral evaluation. So these are data reported in this paper, taken 48 hours post-infections and 72 hours post-infections in presence or absence of drugs or combinations. So many drugs have been today used with the micellar systems for different purposes. But here it gives you some kind of type of data that can be generated. So here are represented here the relative viral productions. So here we fix the 100%, so the one being 100% of viral replications, being with untreated condition. Untreated means that there is only the virus and no antiviral present. Then here are represented in this line the effect of remdesivir. So as you could see very clearly, remdesivir clearly decreases here the number of viruses. So we are talking about logs here. So 10 to 7 logs here of decrease of virus genome in this test. So clearly we can see if a monotherapy is effective compared to this. But also, and this case reports the effect of combinations. If we co-treat remdesivir with diltiazem, so diltiazem, which is a calcium channel antagonist known to induce type 3 interferon response. So alone, it has not so much effect on SARS-CoV-2, but also a small decrease. But now, present in combinations with remdesivir, as you could see here, you could have 1.5 log of differences of decrease. So meaning that in this here, diltiazem enhance the antiviral activity of remdesivir in the system. So you could use here, let's say, this system to discriminate the potency of your compound, either as a combinations or alone. So I'm now coming back to the conclusion. So I hope I have convinced you that air liquid interface 3D human airway models like the mucillar and smaller are useful tool to study SARS-CoV. first for the isolation, characterization, and amplifications of circulating strains, because there would be mutations of the strains. So here we can culture them with these systems, but also for the identifications of transcriptional and secreted innate immune signature due to the SARS-CoV-2, which may help us to find really targeted therapies. And in this context, they would be used for the pathway identification of the pathogenesis. But last but not least, they are today used by many groups and us and many groups as a screening platform to test antivirals. And in first instance, it's for repositioning of marketed drugs, because we are in a situation of urgency. We need to find therapies very fast as we could, or to evaluate how we can use these marketed drugs as, let's say, combinations. But also at the same time, we need also to think about novel therapies and also the context and the mode of administrations of these therapies. And again, these systems could allow us to clearly see if a systemic application would be better or not than, for example, an aerosol therapy. So in this context, locally on the really host cells which are primarily infected by SARS-CoV-2. So I'd like to let's say really warmly thank collaborators, especially the VIRPAS groups in Lyon, led by Manuel Rosa Calatrava, and also Professor Carl, University of Geneva. And I would like also to warmly thank all my collaborators from our French lab at Epitelix and also our Swiss lab. And I would like to thank you all for your attention, and would be happy to try to answer any questions you may have. Thank you.
Great. Well, thank you for that wonderful presentation. We have a few questions coming in already. Just a reminder, you can type your questions in the question box. The first question is about variability. So there are polymorphic variations in the ACE2 receptor, but also human leukocyte antigen that may affect the severity of how people experience the disease. Is this something that could be elucidated with the mucil-air models, given that they're derived from patients? Can you see whether some of them have those polymorphic variations?
So indeed, this is let's say one of the strengths of let's say primary-based human in vitro models in general, meaning that you could indeed see the potential variability of let's say some polymorphisms, some let's say receptor which could be different from a subgroup and potentially within the same pathology, but also interpathologies, meaning that you could reproduce the tissues with, for example, cystic fibrosis patient, but also, for example, with smoker, because it has been described that, for example, smokers could, let's say, be less susceptible to SARS-CoV-2 infection, for example, and better understand that this is indeed something which is absolutely feasible with this kind of models. But to make, let's say, for example, a platform, we have which is not variable and let's say subjected to the inter-individual variability. We have also developed a platform which is a pool of donor, meaning that we mix cells from different donors, 14, and then we end up with a let's say models which let's say would give you let's say a response from a subpopulations, and it's widely used in the context of antiviral screening. Yeah.
Mm-hmm. Mm-hmm. Okay. Just a question about the some of the endpoints in that slide with all the endpoints that you were looking at in the assay. You mentioned one of them was whether the viral material was still infectious. Does that mean that you're testing whether infected individuals would infect others? Is that what that's looking at?
Yes, indeed. Because you can have let's say viruses, let's say, or RNAs that you could detect from the viruses which is spread out and produced by the infected cells, but who could potentially, with the treatment in place, lose their infectivity properties. So indeed, we collect here. Technically, we collect again this virus and we reinfect other type of cells. And we see if they have this potency to infect this other type of cells. But still, it's in vitro with other in vitro systems. And for this, the most known system is the Vero E6 cells.
Okay, interesting. We have multiple questions within a question, so I'll just read them out. So how did you infect the nasal and bronchial tissues? Let's start with that one.
So we used here initially, let's say, a circulating strains which was collected in France, and which was well characterized. And from this, we make some stock of cells because we could, as I mentioned, infect the epithelia. So here we make some solutions of this viruses with a specific MOI. Multiplicity of infections, so which is fixed at 0.1 here. So, but practically, it's a solution of the virus that you add on top of the epithelium.
Okay. Did you wash away the mucin?
Yes. So initially in the test, and this is a good point because you, for example, in the first inoculation period, you add your virus, and then you have to wash off several times to remove the remaining virus, which is apically, to be able to really quantify only the replicated virus afterwards. So in this case, you will wash off the mucus. However, we have also to note that it's the airway epithelium has goblet cells, which are constantly regenerating mucus. So within the test, within 1 week, 1 day, you would have regeneration of the mucus.
Okay. Sort of along these same lines, what was the contact time of the virus in the tissue? So you put the solution on, does it stay there, or do you wash it off after a certain period of time?
So here, initially, the virus for the SARS-CoV-2 is stayed for 1 hour. It depends on the virus. Some virus could have stay for 3 hours, for example. Then you wash off thoroughly to remove everything which was, let's say, stayed apically, and then you can, let's say, co-incubate with the treatment and with different time depending on what is your strategy in terms of administration to the patient. But really fixing, you can really fix to the posology which is targeted.
Okay. And did you mention the clinical strain of the virus that was used, or does it depend on the publication?
We used for SARS-CoV-2 only clinical strain, yes.
Okay.
So meaning that it was isolated in France, and so it was fully characterized, and then yes.
A question, I think, coming back to your response to another question, wondering why Vero E6 cells are used instead of human-derived cells.
Well, this is a good point. We have to understand that Vero E6 cells, which are green monkey cells, are, let's say, the gold standard of let's say, virologists. So when they are thinking about using first let's say infection with in vitro, they would go first with Vero E6 cells because it's very easily accessible. But indeed, so they have a lot of drawbacks. So first of all, they are not human cells, but also they could be used for large screen, for example. But you have a lot of false positive and false negative, which could come into play with their use. First, they have incomplete Vero 6 cells interferon response, such as type 1 interferon is not present in these Vero 6 cells, and this is one of the innate immune defense of the native epithelium in vivo. So for these reasons, you could lack with a response with these cells. And so there is one paper recently published also, for example, in context of testing hydroxychloroquine, where, for example, hydroxychloroquine has found to be positive working on Vero 6 cells. It was found to be not working on the mucillary. It has found to be not working on let's say primates, non-human primates, and it has found to be not working neither in clinical with human. So meaning that, as you could see, the use of Vero 6 cells is questionable also. Yes. But this is we have also to deal that it's really pretty much really quite they are accessible in all virologist labs. Yeah.
Okay. Yeah, that makes sense. Well, that's all the questions we have from attendees. I have one more question that maybe I could get both of you to briefly answer, which is, as we have seen this crisis, this public health crisis happen, I have been really cheered to see all of the human-based models and being used and being published to find therapies and understand the disease. But of course, there is also a lot of funding going into developing mouse models and other animals, using monkeys in research, and a lot of funding going in that direction. So what do you think is one of the main needs that policymakers should be thinking about, not only for this pandemic, but in the future? What should they be doing to try to prepare us for and our public health system for better research now and in the future? Is there maybe one priority that you'd like them to keep in mind?
Shall I go in first, Christy?
Yeah.
I mean, for me, the first thing about this whole crisis is that it's disruptive and it makes people, think about doing things differently. So, I mean, yes, there will be human-based models and there will be animal models being used, but a lot of people will be sitting there. Their lab may have closed down or be running on a sort of skeleton crew, and they're thinking about their next experiment. So it's a good stimulus for new change, but that's very much in the researchers. In terms of policymakers, I think what we've seen is that when you have a panic and people need a fast treatment, we just don't have the techniques to test things out on humans, and so it's only where you've got a drug that's been approved for some other replication you can push it out quickly. So the whole time scale and methodology of testing new therapies, new drugs it needs to be, we need to figure out how to speed it up. And that can only come, I think, from confidence, which is based, I think, on good science. So yeah, it's a very general answer to your question, but I think it's a time to think about the science and think about new approaches.
Mm-hmm. Sam, anything to add?
Yeah, so I fully agree with Malcolm here. And now considering maybe my research area and modeling, let's say lung, indeed, so we are trying to recapitulate here some key modulations of pathologies. So some of them could be nicely captured, but still we are still lacking some interactions with other type of cells, and it's very important now to think about, especially in this context of adding more complex, let's say, immunocompetent models. But still, we need also to think about practical models. So meaning that we still should have all in mind that what we have to develop are something which the other labs should be able to use also in their research. So meaning that it's very important for the community to think about how should we transfer, let's say, our models, how should we complexify, for example, with human cells in context of human immune cells in context of lung, for example, but having models which could be reproducible for the and also transferable, and this is something which also I think for policymakers need to be taken in considerations also.
Sounds like more dedicated funding towards all of these considerations is needed. So thank you both for your work, and thank you all for participating.
Pleasure. Yep.
Thanks for your presentations.
It was a great pleasure. Yeah.
Great, yeah. Thank you. Thanks for your attention, everyone, and we will see you next month.
And can I just ask, Christy, the slides and so on are going to be made available? You've recorded the event?
Yes. Yep, that's right.
Good. Okay.
Take care, everyone.